All right, guys, it's Mr. Brotherton here. We're going to be talking about nucleic acids. All right, I'm starting with this little picture of all of these helixes because this thing is a helix. So this is a similar structure we've seen before. It's how we store carbohydrates to make starch and glycogen, and it's also how we have proteins folded in those alpha helixes. So this is a very common structure in biology. All right, so here I want to introduce you to two figures, one of which you've probably heard of before, and one of which you probably haven't heard of before. So this guy down here is Hans Driesch. He was a German embryologist in the 1800s, and in 1885 he did this experiment where he took a sea urchin embryo and he split it apart. So like all things, a sea urchin is going to start with one cell that's going to divide. And so when it was at the stage of being 4 cells, he split each of those 4 cells apart. And what he found is the process of cell division would actually start over from there, and he would get 4 complete sea urchin individuals. So he actually was making clones of sea urchins by splitting that early embryo apart. And this gave him the idea that each one of these cells has to have some idea of what's going to happen to this entire creature. And the way he put it is that there must be some idea in advance that these cells have in order to make this thing happen. Okay? And up here we have Erwin Schrödinger, who you've probably heard of before as a famous quantum physicist. He had this famous thought experiment about a cat, which you can look up. But in the 1940s, we had pretty much figured out a lot of the fundamental physical laws that are happening in physics, even in quantum mechanics. And so he's kind of started to turn his attention towards what is life. So he was asking biologists, like, what is this stuff? It makes no sense. Okay, it doesn't really conform to a lot of the properties that we know about in physics. There has to be something going on here. And up to that point, biologists were pretty much asking themselves, how does life work? So it's mainly observational. And he was interested in a deeper question, well, what is this stuff? And so Hans Driesch here with his idea in advance, and then going all the way here to Schrödinger in the 40s, who was asking, what is life? What's fundamental here? This goes back to an older idea, the idea of a life force or this vital essence, that somehow life was special from non-living matter, and there had to be, as Schrödinger put it, there had to be some new physical laws happening here. Niels Bohr, another famous physicist, agreed with him, and so did Heisenberg. They all thought that life must have some famous or some special physical properties that makes it different. When really Hans Driesch's idea in advance was this incredible storage molecule known as DNA, and what that idea in advance is basic information about how to build body plans. All right. So what's the function of a nucleic acid? It's to store and transmit hereditary information. So there's no new physical law here. It's just the idea that we can transmit information generation to generation about how to make these special creatures. There's 2 types. There's RNA and there's DNA. So RNA is ribonucleic acid, DNA is deoxyribonucleic acid, and the monomer of DNA and RNA is going to be what's known as a nucleotide. So a nucleotide has 3 parts. It has the nitrogenous base, which is going to be made of these carbon-nitrogen rings. The reason it's called a base is because it's actually a basic thing. So it can absorb a hydrogen atom, and that's one definition of a base, something that can take on a proton. And it has a 5-sugar backbone here. So both ribose and deoxyribose, they both have a 5 carbon backbone. So here's carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5 are right there. And then they also have this phosphate group, which we've seen before when we were talking about phospholipids. So you're kind of starting to see these little repeating building blocks that keep getting used over and over again. So what are the differences between RNA and DNA? Well, RNA is a single nucleotide chain. Okay, so it's more like this. It doesn't have a partner over here to meet up with it. DNA is a double nucleotide chain, okay, so that means its nitrogenous bases actually bond up in pairs and match up with a complement. Okay, it's going to be spiraled in a double helix, like how this is. And that double helix was first proposed as the structure for DNA by James Watson and Francis Crick. Okay, so they didn't discover DNA. All right, I want to point that out. We knew that DNA existed. We just didn't know what it looked like, what it really did, and how it worked. So they were the first ones to propose the structure of DNA. So then there's different types of nucleotides. Okay, so there's different nitrogenous bases that are happening here. So there's purines and pyrimidines. So the purines here have a double ring nitrogenous base. So you can see they have two ring structures in our purines, and those are going to be adenine and guanine. And then our pyrimidines have a single ring nitrogenous base, and that's going to be cytosine, thymine, and uracil. So you can see the pyrimidines have one ring and the purines have two rings. There's a good mnemonic to help you remember this, and that is that the purines are pure as gold. So they are pure as gold. So if you remember that, purine has the word pure in it. As gold stands for adenine and guanine. All right, a way to remember that the pyrimidines have a single ring is the idea that pyramids are sharp. So pyramids cut, or you can think pay cut. So if you remember PY, pay cut, C for cytosine, U for uracil, and T for thymine. Another thing you can think about is if you use pure as gold, gold has a higher value, so there's more rings here in a purine, whereas if you took a pay cut, well, then you're going to have less money, so you're going to have less rings here. So how do we kind of build this polymer from these monomers? Well, we build them 5' to 3'. So what does that mean? We stick them together. We stick our OH group to our phosphate group, and that's how you can get this nice long polynucleotide. So that backbone is made out of what we call phosphodiester bonds. Unlike the other bonds we talked about, this is not a dehydration synthesis or a condensation reaction. Okay, this one does not make water. You're sticking your sugar phosphate together by sticking the new base is going to be added to the sugar of the previous base. So it's going to grow in one direction. So if you look here, here's a nucleotide, here's a nucleotide, there's a nucleotide, there's one, all right? And right here, that's where your bond is going to be. So this is the 3' end of this guy, and this is going to be the 5' end of this guy, where you see that phosphate group, all right? So all of those are showing you where those bonds. So why is this important? What's cool about this is you have all of this hanging off this left-hand side here, and then you have these nitrogenous bases hanging off the other side here, and we need it to be in that shape because of how this thing is used to carry information, and we'll get to that in a second. All right, so how do nucleotides pair up? So remember when I said that Watson and Crick didn't discover DNA? Well, they didn't. We already knew about it, and there was this guy, Chargaff, in 1950, he published a paper about the pairing rules that are happening between nucleotides. So he was looking at different organisms, and he realized that all of the guanine, if you took from an animal, was equal to all of the cytosine in that animal. And he realized that all the adenine in an animal was equal to all of the thymine in that animal. And these became known as the pairing rules. So A is always going to be paired with T, and G is always going to be paired with C. Okay, so if you look here, this A is paired with T, this G is paired with C. What's cool is you can see that it's always a purine pairs with a pyrimidine, all right? And the other thing to know is that A and T have only 2 hydrogen bonds, whereas G and C have 3 hydrogen bonds. So why is this important? Well, a really important thing for DNA is the fact that DNA is a very regulated molecule. It has a very tightly regulated structure. That means it doesn't vary all crazy like how a protein does. And what's going on here with these very strict pairing rules, you can see that DNA is always going to be completely parallel like this. It's not going to bulge or kind of get skinny in some places and bulge in other places, because that could be problematic. Originally, we thought proteins were actually the source of genetic information because there's so many of them. It just made more sense that that would store the information. The problem with a protein is, remember, they have those R groups hanging off of them, and they have a lot of crazy chemical reactions that are happening. So if you needed to read a protein, it's a lot more difficult. Whereas if you think about this guy right here, this is pretty easy to read. It's extremely regulated. It's totally parallel. It's always the same. That's why this is important. So all of that pairing is going to encode information. So it's going to be like the letters of a book, and that stored information is passed from parent to offspring, and it needs to be copied accurately so that way we don't have any mutations that could be deleterious or detrimental. All right. So we call that stored information a gene. So a gene codes for a protein. Okay, so one of the things that's really crazy that we learned about due to the Human Genome Project is that humans actually don't have very many genes. We have a lot less genes than most plants. We only have 24,000 genes, but we have all of this DNA. And each one of those genes stores the information to build a protein, and those proteins are the doers of cells, and they're the reason we all look completely different, because we have different copies of what those proteins look like. All right, so in animals like you and me, we can condense our genes down into something known as chromosomes, which happens during cell replication, and we'll talk about that in a lot more detail later. But just as a quick overview, so here's your nitrogenous bases. This whole thing is a DNA strand. If you take a specific part of this that codes for a protein, that would be a gene. And then we condense these things and we have 23 pairs of chromosomes. Okay, 23 pairs means 23 are from mom and 23 are from dad. So let's get into this. The DNA molecule, what's really cool is it's made of these hydrogen bonds. What do we know about hydrogen bonds? Well, they're not that strong. Okay, so these hydrogen bonds, it's pretty easy to get in there and break them and actually have another molecule come and read the information that's stored in these bases. So that's extremely important. Whereas the backbone of our whole entire nucleotide or our whole entire DNA chain here is going to be made of covalent bonds. So they are not going to break. So in replication, you have these two complementary strands of DNA, and so basically one can be read, all right, and then the other one can be hanging out there to store that information. So it's kind of a copy of the other one, a complementary copy. So you can read one of them, and then if you need to replicate, you can just come in and read the other one. So they can always build the other one at any point in time. So if you split this thing in half, you can always rebuild that other copy just by matching up your nitrogenous bases. It's a very, very good system. So when is the cell going to copy itself? Well, that happens during cell reproduction. So during mitosis, like what was happening with that sea urchin we talked about at the very beginning, that was a mitotic process. So each cell was being replicated. And in meiosis is the other way that cells are going to be copying their DNA. All right. So if we come here, we can look at this, and this is when we finally really realized that DNA was the ultimate source of genetic information. It's because it's such a perfect, good system for doing this. And the first two guys to really make the complete model of what this looked like were James Watson and Francis Crick. And once they realized the elegance of this molecule, this kind of backbone with this very regulated system, it's not bulging and changing shape at all. once they realized that it could do that and that each strand held the information for the other strand and you could split this apart and create new strands, this idea of semi-conservative reproduction, right? So this new strand is brand new. This is the old one. And if you were to cut this, now we have 2 copies, whereas before we only had 1. That's when it really became obvious that this had to be the source of genetic information. This had to be the idea in advance and that life force, that essence that people had been talking about for a long time. So these guys, their work was based on the work of some other scientists. These 2 dudes were not really bench scientists so much as they were really interested in ideas. Okay, I'm not saying that they didn't have the bench skills of a scientist, but they were really idea guys. So in 1953, they published their initial paper, and by 1962, they had won the Nobel Prize. The 2 people who were more of the bench scientists behind their model of DNA were Maurice Wilkins and Rosalind Franklin. So Maurice Wilkins and Rosalind Franklin were both colleagues. They were working on a technique called X-ray crystallography, which is where you shoot X-rays at a molecule. You get these refraction patterns, and then you try and figure out what the thing looks like based on these refraction patterns. So it's a lot of really complicated math, and Rosalind Franklin was a master of this technique. Wilkins was pretty good, not as good as Rosalind Franklin, and kind of what happened here is we think Maurice Wilkins might have been taking her images and smuggling them to these 2 guys, and so she was really focused on getting all the math and everything right, and these 2 dudes were more focused on coming up with an answer, and their answer just happened to be right. She just didn't want to rush to any conclusions until all the math was done, and that was kind of facilitated by this dude right here being an intermediary between these different groups of minds. So interesting note here, the ratio of ATs and GCs is going to affect the stability of the molecule because one of them has 2 hydrogen bonds and the other one has 3 hydrogen bonds. So if you have a lot more GC bonds, it's going to take higher temperatures to break those hydrogen bonds and separate those strands. Whereas if you have mainly AT bonds, it's going to take a lower temperature to break those bonds. So a really cool evolutionary pressure here. If you're an extremophile and you live in a high temperature environment, your DNA code is going to be GC biased because that high temperature could actually break down your genetic code otherwise. Another really interesting thing here is that parasites tend to have more of an AT bias in their genetic code. So this is still in the very observational stages. There's been papers published about this, but we still don't really understand what the evolutionary pressure is there that makes parasites like Plasmodium have these AT bonds instead of GC bonds, which is very interesting. Really, other interesting thing here is adenosine triphosphate. Guess what? This thing is a modified nucleotide. If you look here, you have a phosphate group, you have your ribose sugar, and you have a nitrogenous base here. So adenosine triphosphate. So that thing that gives you energy is actually a riffing on DNA or RNA. By just adding some extra phosphate groups here. Another cool thing about adenosine triphosphate and caffeine is that caffeine works by blocking binding sites for adenine. So if you have a bunch of adenine floating around in your brain, it makes you feel tired because you've broken all these phosphate groups off and you just have this dude floating around. So your mind says go to sleep, tells your body to go to sleep. Caffeine actually blocks those. So caffeine does not give you energy. It's a biological trick. And we'll review these tomorrow in class. We'll do the macromolecule review tomorrow before our test. Okay, so that's a quick little rundown of what's going on with nucleic acids. Make sure you know the differences between DNA and RNA, and we can talk a lot more about what's going on with RNA as far as its role in the replication of DNA and its role in building proteins, all right? Because RNA is a really cool little molecule because RNA can actually act like both a protein and it can actually store some genetic information. And it might be possible that in the history of the world, the first replicating molecule that stored information was actually RNA, not DNA. And we get that evidence partially from viruses that are RNA today. But we will get into all that stuff in class. All right, guys, thanks a lot. That's all I have for you today, and stay curious. Bye.